Everything is personal right here. Everything is personal right here.
Ladies and gentlemen, boys and girls, welcome to another episode of Everything is Personal. And today, our guest is Dr. Bill Andrews. And this is the last time I'm going to call you doctor for the whole show. Welcome, Bill.
Thank you. Thank you.
So tell me about this. We were talking earlier, I cut you off. You didn't want to be a doctor or to be handed you a PhD. I want to be handed a PhD.
Yeah. Well, my goal, my entire life has been trying to figure out what aging is and why we age and how we age, and more importantly, how not to age. And so, in looking at all the theories and things like that existed, I started realizing from all my other studies, like in logic and common sense and statistical theory and things like that, I started realizing they don't make any sense, well, including biology, especially. And so I just was studying everything I could to figure out what is causing us to age, because we'll never know how to not age unless we know why we age and how we age, and so and what aging is too. And so I was just studying all that and going to schools and taking classes and stuff like that, and I got a PhD. It's like I never had that as a goal. I just wanted to know everything I needed to know to cure aging, because it's been an obsession of mine since my father's. It was an obsession of my father's, and he passed that on to me when I was 10 years old. He and I teamed up together for years.
I was wondering, I think you just kind of answered the question, but what was the defining moment when you said, all right, well, curing aging is my mission? And in curing, it's an interesting approach. You hear people like David Sinclair, and they talk about Dr. David Sinclair. They talk about, the disease of aging, but we were never taught that way. It's sort of the circle of life, right? You're born, you live as long as you're supposed to. I know genetics. So, all right, you have a genetic predisposition, and people for years are like, it's your genes. That's how long do your parents live for? I was like, no, I mean, that's epigenetic. You can go and do something about that. So that's as far as my knowledge goes. And then, they talk about senescence and all, and we'll get into all that stuff. But curing, that's an interesting word.
Well, I didn't realize that was an incorrect word by some people, considered by some, until like 1990s. Okay, because when I was 10 years old, and my father came out to the front lawn and talked to me, because I was out there with my 4-foot, 8-inch wide reflector telescope, looking at the moons of Saturn and the moons of Jupiter and the moons of Saturn, rings of Saturn. Sorry about that. I was just, he knew I was obsessed with science and medicine, and that was pretty unique for somebody at 10 years old to be doing that. So he came out to the front lawn and he just said to me, and this is word for word, I'll never forget it, Bill, when you grow up, since you're so interested in science and medicine, when you grow up, you should become a doctor and find the cure for aging. I don't know why nobody's ever done that before. Okay, that's what he said. Now, he was a television producer, so he wasn't a scientist. He and I would argue over what's the molecular weight of water. He would say it was 2H2O. No, I'd say it's H2O. The 2 just means there's 2 of those molecules. Those are the kind of, that's where his science was. But he really loved living, and he just couldn't take this idea that he is eventually going to die of old age. And he did. He died 10 years ago. No, 10 years ago from Alzheimer's disease. And so I lost my best partner then, but he's the one who gave that idea to my head. But I never thought cure means disease until everybody's suddenly in the 1990s, when I was speaking at conferences and things like that, people would say, you can't call it curing aging if it's not a disease. I said, what, aging is not a disease? And I said, what's going on there? And so that just increased my studies even more. And I'm convinced that aging is a disease because I understand the evolution. And part of my PhD, when I was getting my PhD, it was studying not just biology, but also evolution, because I wanted to know, if aging is so bad for us, why didn't we ever evolve a way not to age? So I spent a lot of time working on that. And I ended up learning a lot about evolution. And as a result, I ended up really getting a really good understanding of why we age. And because of why we age, it is a disease because it had to be an event that was created in our bodies. Okay. before sexual reproduction, life did not age. There was no aging processes in life. Okay. It was later on, shortly after sexual reproduction evolved, aging became something that was advantageous to the species, not advantageous for the individuals, but advantageous for the species, because by eliminating the longer lived that had already raised their young, that increased the chances that the offspring would interbreed, as opposed to the offspring or the parents rebreeding with the offspring or themselves. And by letting the offspring interbreed, that increased the diversity within the species, because sexual reproduction is all about gene shuffling. And gene shuffling didn't really exist before sexual reproduction. So now the diversity is increasing a lot from sexual reproduction, but only if it increases even more if you eliminate the longer lived and prevent them from rebreeding. And that increases the chance that our species would survive rapidly changing environments. So that's why that happened. But many life forms have evolved an aging process by different mechanisms. Okay. And so the humans, we have a whole bunch of different aging processes. And I like to think of them as multiple sticks of dynamite that are burning inside of our cells. So the question is, which stick of dynamite has the shortest fuse? And that's the one we have to work on. And in humans, that stick of dynamite with the shortest fuse is, without a doubt, telomere biology. Okay, telomeres shorten. I haven't described what telomeres are, but that maybe I should go into that right now. I want to preface that I'm not a person that's totally sold on telomere biology. I'm interested in curing aging, whatever it takes. But I ended up realizing back when I was still in college that, or actually, I realized something else in college. But what I realized later was that no matter what else we do to try to cure the aging process, aging will never be cured unless we also solve the telomere shortening problem. And it's the only theory, and I don't even call it telomeres a theory anymore. It's the only, I'm going to, for a minute, it's the only theory that has that hard roadblock, okay, that can be defined that way. But none of the other theories can say, well, we can cure aging even if telomeres shorten. Okay, none of them do. Okay, because telomere shortening is a thing that's well known in every lab that ever works with human cells. They know that human cells can only divide a certain number of times. So let me explain what a telomere is, unless you want to interrupt. Okay, so telomeres are found at the very tips of our chromosomes. Our chromosomes are where our genes are. The chromosomes have this long string of beads inside of them that are bases. I'm sure you know all this, but maybe somebody in your audience doesn't know this. But the beads are the sequence of the beads are what encode our genes. And our genes are what give us our hair color, our eye color, things like that. And they control a lot more than that. But the beads, we have like 23 different beads and string of beads in our cells. And think of them like shoelaces. Okay. There's 100 million beads. The beads are also called bases in each chromosome on average. At the very tips, there's something equivalent to what we have on our shoelaces that are called telomeres. So on the tips of our shoelaces, they're called aglets, and they protect our shoelaces. But our telomeres, our chromosomes, have the same thing. They're called telomeres, and they're at the very tips of our chromosomes, and they protect our chromosomes from degradation from the ends, but they also play a role in the regulation of genes on the chromosome. Every gene has like a dimmer switch next to it that turns it on or off at different levels. And telomeres actually can control the dimmer switches on those genes that play roles in aging. In fact, almost all the other hallmarks of aging have been shown to be under the control of those telomeres, the dimmer switches that control those genes. And so the problem is that because our species wanted to survive, and I don't want to say want because that makes evolution sound like a force when it's actually a result, but sometimes it's easier to talk about evolution as a force. But we evolved a way to eliminate the longer lived, as I said, to to increase the chances that our offspring would interbreed. Now, that meant that the way humans did that, and not only dogs, cats, horses, sheep, pig, and deer are the only other animals that have evolved this aging process, and that's telomere shortening causes us to age, and therefore the way to make telomeres shorten was to get rid of an enzyme, turn off an enzyme gene called Telomerase. So when I first, I've known since the 1960s, and at my anti-aging clubs in high school and college, we would discuss the fact that it had been well published that Dr. Leonard Hayflick had discovered that human cells can only divide a certain number of times, and then they stop. And we thought, well, this must have something to do with aging, because no matter what else we do to solve aging, aging is still going to happen if we don't solve this limited number of cell divisions that humans can do. So we would sit and rack our brains trying to figure out how could a cell know how many times it's divided and how many times it has left to divide. And we came up with all these kind of theories, but most of them there was no mechanism in biochemistry to explain it until we came up with the idea of ride tickets at an amusement park. And so we used to talk about there's something that's like ride tickets at an amusement park inside of our cells. Every time a cell divides, it loses a ticket. When it loses its last ticket, it can't divide anymore, just like you can't go on any more rides anymore when you use your last ride ticket. So the aha moments were first when my father came out to me and said, Bill, when you grow up, cure aging. The other one was like years after we came up with this ride tickets at an amusement park analogy, when we couldn't figure out anything in the cells, what those were. In a conference in 1993 in Lake Tahoe, I was sitting there and I heard a guy named Dr. Calvin Harley say that every time a cell divides, the tips of your chromosomes get a little shorter. And he said that he can measure the length of your telomeres, and he can tell you how old you are and how long it'll be before you die of old age. I was blown away because here I'm sitting there thinking, these are the ride tickets. I never, ever heard about telomere shortening like that until that moment. So before he could even get off the podium, I was at the bottom of the stairs of the podium, asking him, has anybody figured out how to re-lengthen or add, I didn't use the term ride tickets, but how to add ride tickets back? And he said, I'm working with a company called Geron Corporation. We've been working on this for years. We've been collaborating with labs all over the world. Nobody has figured this out. And so I just boldly said, let me come and work for you and I'll figure it out in 3 months. Okay. And, shortest job interview, he offered me the job. And the reason was because I had already been in this time when I was trying to figure out what these ride tickets are. I was working at biotech companies doing cancer research, heart disease research, inflammation research. I'm one of the co-inventors of human growth hormone, tissue plasminogen activator, beta seron, thrombomodulin, renin, all kinds of things. I was awarded National Inventor of the Year for my cancer research. So I was a walking perfect resume. Okay. So he thought, well, if anybody could do it, I could do it. And then, so he offered me the job. I quit the company that I said I was a lifer at because I loved the company called Burlex Biosciences. And I went to work at Geron Corporation, and he put 3/4 of the company, of the scientists, reporting to me. And 3 months and 17 days later, we discovered the enzyme human telomerase. And the extra 17 days was because they were
You need to write about it.
interfering with me too much. It was like, and I blame them for the extra 17 days. But
I wanted to ask you a question before we move off of telomeres. So, the shoelace phrase at the bottom,
yes.
and I wonder if the telomeres do the same thing. And do every single time the cell divides, and I know maybe this is way too inside baseball for people, but I don't care. I'm talking to you, so I'm going to pick your brain on this.
Right.
When a cell divides, do we know how much that telomere actually shortens every single time?
Yes.
Or is it's predictable?
Oh yeah, we know it's not 100% consistent, but it's average of about 40 bases. So
Interesting.
40 bases is equivalent to one ride ticket.
Mm.
We're born with telomeres that are 15, or conceived at telomeres that are 15,000 bases long. What is it? 15,000 divided by 40. That would mean, well, that's 100 ride tickets, because the 5,000 bases closest to the chromosome itself don't count. Okay, so it's really, so the telomeres are 10,000 bases divided by 40 or 50. Let's make it so that that gives us about 100 ride tickets then. But then there's so much cell division from going from a newborn baby to from a single cell embryo to a newborn baby, that we've already used up half of those tickets just to be born. So the rest of our life, we can use up. I did get the math wrong there, didn't I? So the math there is that we have 100 ride tickets left when we're born, not when we're conceived. And so it's when we use up those tickets, yeah. So then our cells lose the ability to function and we die of old age. But even during the process, I want to go back to where we were talking about telomeres, but let me finish this one first. Telomeres also play a role in, as I said, turning on the genes by folding over. They're like enhancer sequences, if you know what an enhancer sequence is, that actually fold over, and the chromosomes fold over, bring enhancer sequences to the dimmer switches, the promoters, and turn them on and off. Telomeres have all the hallmarks of enhancer sequences, and they have proteins that bind to them. It's just they're exactly the same kind of proteins that bind to enhancers. So these telomeres fold over and regulate the dimmer switches, but they become less able to do it when the telomere gets shorter and shorter and shorter. So that's why we see the ongoing symptoms of aging instead of just this total shutdown when telomeres get really short. So even as you're losing ride tickets, you are affecting the rides, essentially. So it's not 100% analogous to ride tickets at an amusement park. But I was going to say that we discovered telomerase at Geron Corporation by considering the fact that if every cell in our body had telomere shortening when they divided, then how could our children be born with longer telomeres than we have? Okay, because our reproductive cells are involved in cell division. The cells that produce our sperm and eggs, they have to go through cell division in the primordial germ cell line. They have to go through cell division. And so why is it that our children are born with the same length telomeres that we had when we were born, but longer telomeres than we had when we were making them or begetting them, or whatever the word is? And so we decided reproductive cells have to have some mechanism to prevent the telomeres from shortening. And so that's what we studied, and we ended up discovering this enzyme human telomerase in our reproductive cells, and it wasn't produced in any other cell of our body. And so the first thing we did was we took the gene for telomerase, put it into a regular skin cell in a culture in a petri dish with the dimmer switch turned all the way on. Okay. And lo and behold, those cells had not only did they stop aging, they had reversal of aging by every method of measurement of aging that we had. Okay. So this made headline news back in the late 1990s or mid to late 1990s, because everybody was saying it's the cure for aging. But I still thought there was a long way to go yet. Now, so we did a lot of research. And so one of the questions you were alluding to was the fraying of shoelaces. Okay. So telomeres do shorten by degradation or fraying or all the different words that are used, but it's not a major part of telomere shortening. But you can. So anything where unhealthy lifestyle or poor genetics can cause what I call accelerated telomere shortening. But the basal level rate of telomere shortening is caused by something called the end replication problem. And that's actually a scientific term, end replication problem. Every time our cells divide, every time a cell divides, everything inside that cell needs to be duplicated so that the daughter cells are identical to the parent cell. Well, that includes duplicating the chromosomes, the DNA. So the DNA is duplicated, but the cell lacks the ability to duplicate all the way to the very end. The analogy I like to use there is like, think of the top row of bricks on a brick wall as being your chromosome, and now making a new row of bricks on top of that is like duplicating your DNA. But now you're a bricklayer, you're on top of that wall, walking backward and laying a brick every time. You take a step backward, lay a brick in front of you, step backward, lay a brick in front of you. When you get to the very end of that wall and step backwards, you fall off. And you can't replace that last brick. So the new row of bricks is shorter than the original bricks. So that's actually more accurate than you would imagine. Okay, exactly why our telomeres shorten. But that wasn't wear and tear. It wasn't fraying. It wasn't degradation. It was simply the lack of the ability of the cell to replicate all the way to the end. So the old chromosome is still at the same length. The new chromosome is shorter.
So can you have lifestyle modifications that address both of those? So there are certain things you do in lifestyle that slows down the degradation of the telomeres versus these are lifestyle choices that'll accelerate it. I know we still have this many ride tickets. I get it, but can I, this ride is going to cost you 5 tickets instead of 1 kind of thing. So you kind of age quicker.
Well, that's what I'm doing. Okay. So for starters, we do know that oxidative stress, free radicals, like the sunlight, hitting your cells, UV light, all those kind of things do cause damage to the telomeres, and therefore they accelerate at a faster rate. Okay. Obesity, smoking, a whole bunch of different things. So you can reduce the rate of accelerated telomere shortening by antioxidants, quit smoking, don't be obese. And there's also a factor called exercise, which I'll come back to in a second. But
Yeah.
I wanted to say that I'm trying to think. So there's also inflammation, which I mentioned before.
Yeah.
But inflammation isn't the degradation. That's just increased rates of cell division. So you can also do things to decrease the rate of cell division.
Yeah, because if you're increasing the rate of cell division, you're using more ride tickets in that way,
Yes.
correct?
Exactly. Now, things that people do that accelerate cell division include like microdermabrasion on their skin. It includes drinking alcohol, drinking alcohol, anything that, well, drinking alcohol kills liver cells. We all know that.
Yeah.
But that's okay when you're young because you have other cells in your liver that would divide to replace those killed cells. Okay. But now because you're drinking, you're accelerating the rate that cells have to divide to replace killed cells, and therefore you're accelerating the rate of telomere shortening. And so when you run out of ride tickets in your liver, you suddenly can't replace the dead cells anymore, and the dead cells just accumulate. That's what liver cirrhosis is, and that's what kills you. So if you could stop drinking and quit killing your liver cells, you would live longer. And the same as smoking in your lungs, same with muscular dystrophy by the mutation of the dystrophin gene. There's all kinds of things that will accelerate cell division.
Well, what about food? Like, what about certain types of foods that you will consume? Is that something a contributor?
Yes. Yeah. Well, I mean, there's a lot of, I don't want to say politics in the food, but I'm personally
just you and I are just talking. Nobody's listening. Doesn't matter.
I believe in a vegan diet, and I also believe in no oils, based on
no oils at all, or no seed oils.
No oils, not even olive oil. Okay,
really?
Yeah, and that's based on studies by Dr. Caldwell Esselstyn that I've read, and my critical meta-analysis of his studies pretty legit. But it's something that oil will close up your collateral arteries, and make it harder for you to pump blood to your heart when you need it. And so, since I'm an ultra marathon runner, I want to have as much blood going to my heart as possible. And so I restrict oils from my diet, because there are people that have their coronary arteries 100% closed and live perfect lives because the collateral arteries are flowing the blood, and collateral arteries don't have plaque buildup and stuff like that, like the coronary arteries do. So by the way, I'm one of the inventors of tissue plasminogen activator I mentioned before, which is something that breaks up blood clots that close up the coronary arteries. So that's my diet. But
I'm sorry to interrupt. I just want to go back to that for 1 second, because I don't eat meat. I haven't eaten meat in a very, very long time. I eat fish, and one of the reasons why is omegas, right? You need omega-3s to feed your brain. And you're right about plaque. And by the way, we've done clinical studies about amyloid plaque being associated with Alzheimer's as well, right? So galactin-3, et cetera. So what you're saying makes sense, but like, all politics aside, you have this blue zone and diets, and they consume a lot of oils and olive oils and all that stuff. And avocado is a really good fat, and you can consume that oil. So if I'm consuming fish oil or fish to feed my brain, let's say, do I not follow these blue zone diets of longevity where people live in 100+ years? Are we contradicting that?
No, I mean, the main thing that you want to avoid when you go on a vegan diet is arachidonic acid. And that's what's his name? I can't remember the name of the guy who did a lot of work on that and published a book on it. But arachidonic acid is very inflammatory, and it's found only in meats and fish and fowl and dairy. So you eat plants, you don't get arachidonic acid. And arachidonic is a major inflammatory chemical. But when you purify omega-3s from fish, you're probably not getting very much arachidonic acid.
Yeah.
Now, that said, there are synthetic forms of omega-3 fatty acids called Vasipa, VASECPA or something like that. And that's what I take. Okay. I think it's VASCEPA. That's what I take. Just in case there's arachidonic acid in the omega-3s. Some are pure, some are not pure. So omega-3s and vitamin D, those are probably top two things people should be taking. And both of those actually reduce accelerated telomere shortening. So I speak very highly of omega-3s and vitamin D, and I even talk about the doses that, I believe that we should be taking 1.2 grams of EPA oh no, sorry, 1.4 grams of EPA and 1 gram of DHA every day. And we should also be taking 10,000 IUs of vitamin D every day. Those on the original papers showing that they reduce accelerated telomere shortening, those were the optimum doses. Okay, so that's what I take every day. I take 5,000 IUs of vitamin D every morning and at night, and I take 500 grams of Vasipa every morning and every night.
Got it. Yeah, I take vitamin D3 as well. The only question I always have is like, how bioavailable is that vitamin that I'm purchasing? You know, how do I know that I'm getting those 10,000 units? Is it actually uptaking that much? Who knows?
Yeah, well, usually that's already figured out when they put that on the label. Okay.
We hope.
And so, yeah, well, go to a company like Life Extension.
Yeah.
Okay. Now you can believe everything that they say. And I buy most of my stuff from Life Extension.
Okay.
The real common ones. I mean, there's some companies that I think are really good that sell specialized supplements, and I buy from those too. But the common ones are at Life Extension. I
want to go back for a minute and kind of shift focus back on this longevity and lifespan and health span. It's such a buzzword right now. I wanted, like, you talked about the sexual reproduction in evolution that's creating that, but you have mammals in the world that live for several hundred years and longer. So do they have a different evolutionary process in sexual reproduction than we do? Do they have telomeres as well? Are they different lengths? Like, how do we, if we know that it exists, why can't we replicate that in human beings? At least to start with,
we are
a couple hundred. Okay.
We are. The reason what's been found is like the animals that have been shown to have undetectable aging are lobsters, humpback whales, tortoises, some fish, some birds, clams. Every one of these animals have been shown to have telomerase produced in all their cells.
Interesting.
They have no telomere shortening. They have no detectable aging. They also rarely get diseases, including cancer. Okay. And so that's what I'm trying to do. I'm trying to make us like them. And now mice and all rodents, they also produce telomerase in all their cells and don't have telomere shortening, despite all the studies you'll hear about people delivering telomerase to mouse cells and showing an extension of lifespan. I don't know. I can't explain what's going on there, but we don't, it's not very much extension of lifespan. It's just a short bit. But mice age mostly by oxidative stress and mitochondrial dysfunction. And they have humans have 100 times the level of antioxidants, natural antioxidants, than mice do. So that's why we're not susceptible to oxidative stress like mice are. But as I remember those sticks of dynamite I was talking about, when we put out this fuse from the telomere, okay, shortening, that next stick of dynamite might just be oxidative stress. So we have to solve that problem too. Now,
so do they produce less ATP and their mitochondria doesn't produce any more cells at that point? So that
by they, do you mean mice?
Yeah, mice.
They might produce more ATP because they're going to be producing more free radicals.
I see. Got it.
And so through the process of glycolysis and Krebs cycle and oxidative phosphorylation, stuff like that.
Interesting.
There's a whole subject I could talk about too, but whatever. But decrease that fact. This is maybe a good time to talk about exercise.
Yeah.
Okay. We talked about. I said exercise can reduce the rate of accelerated telomere shortening and aging, but exercise is kind of weird because too little exercise can accelerate aging, and too much intensity in your exercise can increase rate of aging. And it's the reason why I'm trying to connect the dots of what we were just talking about before. But when you run, I just want to say so sugar is really bad for you. Sugar induces inflammation. So does insulin. But when you run or do any endurance exercise, even kayaking or bicycling or anything like that, your muscle contractions induce the production or movement of a receptor called GLUT4, GLUT4, which is a glucose receptor, glucose transporter receptor. Moves it to the surface of the cells, and that's how your sugar gets into the cells to be consumed. Now, when you're not exercising, the sugar goes into your pancreas through another receptor called GLUT2, and that produces insulin, and insulin is really bad for you. Insulin then goes, and because that causes inflammation too. Insulin goes to your other cells and induces the production of that same receptor that exercise will induce, called GLUT4. So people need to make a choice. Do they want their GLUT4 to be produced on the surface of their cells from exercise or insulin? And because insulin accelerates aging and things like that. So if you're going to eat a lot of sugar, do it while you're running. That's the.
But I wanted to ask you, Bill, about you mentioned something about not enough and too much. Is that a personal thing? Like, what is too much and what is too much for? Because, like, if for me, maybe I go to the gym. Let's say I'm lifting 100 pounds. Okay, well, if I'm lifting 100 pounds, I'm 175 pounds. So it's not my whole weight, but there's guys who are my weight who are lifting double what they're lifting. So how do we know what's too much, or are we just listening to our bodies?
Well, let's say bodybuilding, lifting weights is a different subject than endurance. And let me come back to that. But in endurance, if you are one of these runners, let's use running example. If you're one of these runners that only runs every 2 weeks and then enters a marathon, you'll do well. Okay, you'll do well. But you'll end up on your hands and knees throwing up afterwards. You'll be stiff as a board from inflammation for 2 weeks, and you can't do anything. Okay, that's what I call about being too intense. Okay, even though running every 2 weeks is still intense. The intensity of running that marathon, because you're really pushing it.
Mm-hmm.
If you run every single day, be consistent. You don't get inflammation. Okay. And you can run these races, but keep it fun. My rule is always keep it fun. When it quits being fun, quit, save it for another day. And after a while, you're going to find out you can run 100 miles easily. Okay. And I know a lot of people that have taken that kind of idea and found it, wow, they're suddenly ultra marathon runners and they thought they could never do it. But that's because they kept it fun.
But is that in okay, so that's cardio and a bunch of other different things. But what about resistance training? Because if you're going to live a healthy life, you need to have a little bit of muscle mass. And
resistance training is okay.
Okay.
I'm fine with the resistance training. It's the bodybuilding. When you're trying to rebuild up your muscles really big to look like Arnold Schwarzenegger, that's an example of accelerated cell division. Because the way you build muscles is to tear your muscles apart from the weight. And then other cells have to divide to fill in those gaps. Okay. And therefore your muscle's bigger when those cells fill the gaps. And the cell division causes telomere shortening. And so, and even though I'm one of the inventors of human growth hormone causes accelerated aging too, because it induces cell division. And a lot of microdermabrasion, immune stimulators, everything, all those things induce cell division, which causes accelerated telomere shortening, which causes accelerated aging.
Yeah, I see those guys at the gym. I mean, it's pretty clear and obvious. I wanted to ask you about, am I pronouncing it right? Telomeres?
Telomerase, but I pronounce it telomerase.
Telomerase.
I also pronounce telomeres. So it's just kind of contradictory, but every pronunciation is correct.
So I wanted to ask you, I'm going back to what you mentioned about the skin. right? So you were able to put the telomerase on the skin, and it reignited or re-energized the aging process, actually reversed aging of that skin. So if I'm kind of, really quick, short line and straight, which it never is in science, that means that all I have to do is apply telomerase therapy to my cells, and then I'll re-engage them and extend my telomeres, and then start all over, right? Isn't that a good approach to anti-aging therapy?
But how do you do telomerase therapy? Okay, so
That's what I was going to ask you.
telomerase has to be inside the cell.
Right.
Okay, and telomerase is too big, so you can't just spread telomerase on your skin. But there is something called gene therapy, okay, which is a way of delivering a new gene to the cell with the dimmer switch turned on.
So like CRISPR?
Well, CRISPR is an gene editing.
Okay.
And it still has to be delivered to the cells. Okay. And CRISPR is, I should say, I've been doing gene editing for, I want to say 30 years. CRISPR is not the first thing that allowed us to do gene editing. We were doing zinc fingers and then talons. And then now CRISPR's just gotten, for some reason, really great marketing. And I was hearing some noise just now. Sorry, I got distracted by hearing some noise. I thought but a jet. I'm right across the street from the airport, and some jet is racing. What were we just talking?
We're going to telomerase therapy. You were saying that.
This is really important. There is some clinical studies being done right now of delivering the telomerase gene to cells with the dimmer switch turned on. And people are being tested right now, mostly in safety studies, and the place to go is a company called BioViva, BioViva. And it's run by Dr. Elizabeth Parrish. I'm sorry, not doctor. She's a MBA, Elizabeth Parrish. You might want to have her on your show. I mean, she's a very, very interesting person. But it's a very risky at the moment, still in clinical studies, a lot of safety studies being done. And through FDA processes, this, even if it works, it won't be available for 25 years, things like that. But so what I'm doing instead, and I should talk about this because I think one of the companies that sells one of the products I invented is partially responsible for setting up this podcast. I'm searching for small molecules, either found in plants or drugs or things like that, that can get inside their cells and turn that dimmer switch on. Okay. And all it has to do is bind to the protein that's shutting it off. It dislodged that protein, which is called a repressor protein. It's got to bind to that protein in a way that causes the protein to fall off the dimmer switch, and the dimmer switch will turn on. So that's what I've been doing for the last 20 years. And we have like $1 million robots that can test 4,000 different chemicals or plant extracts a day. Plant extracts are my favorite because we don't have to wait the 25 years for them to get approved. But we have, we've tested over 20,000 different plant, and I want to say fractions of plant extracts. We don't do whole plant extracts because a whole plant extract has 200,000 different chemicals in it. Okay, and maybe I shouldn't use the word chemicals because a lot of people don't like that. Molecules. It has 200,000 molecules in it. So we fractionate it into molecule sizes, different molecules based on size or charge or solubility, things like that. And so we'll test like 20 to 30 different fractions of a plant extract every time. And we have found, out of testing 20,000 plants so far, we have found 40 to 50 different fractions of plant extracts that will actually turn on that gene in all of our cells. Okay, and because our mission at Sierra Sciences here is to find something that reverses aging, to cure aging, not just slow it down. We kind of put those plant extracts on the shelf so that we could spend more time testing for more to find something more and more potent things. But
a couple questions on there. So the biggest obstacle right now is the method of delivery, right? So you're trying to
For
find
gene therapy, for
for
gene
gene
therapy.
therapy, right? So, there are things that I read like nanobots, and there's things like plant medicines. If, for example, like phytocannabinoids that have receptor binding to your endocannabinoid system, CB1, CB2 receptors. If you're already able to ingest a specific, ratio, can you use that as a carrier maybe for gene therapy or
But
nanobots or other things like that?
those are pretty well under hand. I mean, the problem with gene therapies are some, okay, gene therapies use viruses or they use simple DNAs. Okay.
Right.
Well, simple DNAs is something I've been doing since the 1970s.
Mm.
Okay. So, but by using a virus, you can get a lot more into the cell and there's all kinds of carriers, lipid nanoparticles, things like
Right.
that can help DNA get into a cell, but nothing as good as viruses can get into cells. Now, contrary to popular belief, a lot of people are promoting the DNA approach instead without using the virus, trying to claim that it's as good as the viral gene therapies. But we do both here all the time in my lab. By the way, behind me is 10,500 square feet of lab space. Okay. And so these are kind of studies we do all the time, but we are always better off with the viruses. Now, the problem with the viruses is that some of them integrate into the chromosomes, which can cause cancer. And that's why a lot of clinical studies that have been done in the past, people died because the principal investigators, and I'm going to blame the FDA, weren't smart enough to know that that will happen. Okay. People like me were appalled. We knew that you take a virus that integrates into the DNA and use that as a gene therapy, 2% of the patients are going to die because it's going to integrate into a gene that's going to, and into a piece of DNA that turns on the telomerase
cancer,
gene,
right?
or it turns on cancer genes or turns
Yep.
off cancer genes. Or cancer suppressant. I should mention, I was National Inventor of the Year in 1997 for my cancer research. So I understand cancer a lot, but the FDA should have known this too. Okay. But okay, so the other problem with a lot of the viruses is they induce immune responses. Okay. But there's some.
But you can do immunosuppressants as part of that,
Yeah,
right?
you can do immunosuppressants, and there's all kinds of different things. You can use decoys, like
Yeah.
first there's also inside your blood, if you've already been exposed to the virus for one reason or another, you'll have neutralizing antibodies and also other things called neutralizing substances.
Uh-huh.
So you inject the gene therapy into your blood, it's destroyed before it can ever do anything.
Yeah.
So there's all kinds of problems there, but especially BioViva. Okay. BioViva has come up with some really great ideas. And something that they're pursuing right now is using the CMV virus,
Okay.
which doesn't really induce immune responses, does not integrate, and has another advantage that most of the other vectors don't have. It can carry really large genes. And in fact, so large that it can carry more than one gene at a time, okay, into your cells. And so the stuff that BioViva is working on is really incredible. And so I definitely recommend.
It's so mind blowing to me because, there are two parallels right now. There's this whole notion of longevity, right? We have conferences on longevity. We have all these different things, but the ability to live healthier lives, like it shouldn't be mutually exclusive. It should be the same thing. So I wanted to ask you, is there an iterative process, meaning that are we saying that within the next X amount of years, our goal, maybe we can add another 20 years to our lives, another 40 years to our lives. But when I say years, I don't mean I had an aunt that passed away last year. She was 102 years old. The last 10 years of her life was existing. Who wants to be in a diaper and get their, ass wiped by somebody else and all that? You want to live as you're living now, kind of thing. So is that a possibility in the near future?
Yeah. I mean, I'm sorry I'm talking so much and going off on tangents, but that's really where I want to get to. And so let's talk about the most exciting thing that's going on right now, in my opinion. Okay. And that's this product that just got released that I invented called TeloVital. Okay. It's a mixture of our very top plant extracts, fractions of plant extracts that we discovered that induce the production of telomerase. And so the when you take this, it's the only, it's like you can do other things like reduce accelerated telomere shortening, which will still put your maximum lifespan and health span at 125 years. That's when we do the math. I forgot to mention that before. The telomeres give us an absolute maximum lifespan of 125 years. TeloVital can make us exceed that. And even though, but only if you have the perfect lifestyle and the perfect genetics in addition to that, but it's still going to allow you to live longer than you would have without it. Okay, because TeloVital is inducing telomerase, is inducing these cells to produce telomerase to lengthen telomeres. Now, that said, I got to mention, it's not going to reverse aging. Like if when we put telomerase at high levels into regular cells and skin cells growing on the back of mice and things like that, we saw reversal of aging.
Yeah.
Okay. But now, in the case of something like TeloVital, it doesn't induce enough telomerase to win what I call the tug of war between shortening and lengthening. Okay, so in our reproductive cells, we have a tug of war. A cell divides, the telomeres get shorter, then telomerase re-lengthens it. It divides again, telomeres get shorter, telomerase re-lengthens it. In all the other cells of our body, we only have the shorteners. Okay?
Yeah.
So now what we have done with TeloVital is we've added people to the other side of the tug of war, but not enough to win the tug of war, but definitely slow it down. Okay, so cell divides, telomeres get shorter, telomerase lengthens a little bit.
Yeah.
Divides again and lengthens a little bit. So we do have a decrease in the rate of shortening, but now it's decreasing the rate of what I call basal level telomere shortening, or the end replication problem, not the accelerated telomere shortening. So combination.
How do you consume TeloVitals? Is that a
I'm sorry, I should have a.
Yeah, show it. Bill.
I have an old bottle of it here. I meant to bring. Okay, so this is it right here.
Yeah.
But it. Let's see, can I get the label in there?
Yeah.
It's the thing that this bottle is missing is this was like one of the first ones launched. It's missing that it's now approved by the USDA. Okay, and the newer bottles have that. And as long as I said that, I hope Touchstone Essentials is the company that markets this.
Yeah.
And that's what I was saying is we were putting these things on the shelf, and Touchstone Essentials came to us and said, we'd like to market some of those things, even though they don't reverse aging. We think slowing down the aging, lifespan and health span, increasing lifespan and health span is good. And so I said to him, well, we have talked to other companies. In fact, some other companies did market it, and it was their number one selling product with weaker telomerase inducers. And that includes a company called Isagenix, for instance. They launched product B that later became Isagenesis, but they quit selling it because the profit margin wasn't high enough. So I told the president, his name is Eddie Stone, told the president of Touchstone that, be aware that other companies have done this, but the profit margin wasn't high enough, so they quit selling it. He says, I don't care about profit margins. Okay, well, he became my best friend instantly.
Yeah.
Okay, because I'm only interested in making the world happier and healthier. I'm not in it for the money whatsoever. Yeah. And he told me how he's the same way. And now, so I'm a big fan of Touchstone Essentials. But they're launching this product. They launched it several months ago. They launched it even though they know they're not going to make a lot of money off of it, just because it's really good for people.
Oh, yeah.
Are
there any adverse events associated with that you're aware of?
Not yet, but everything. There's not a single chemical or plant extract that somebody in the planet isn't going to be sensitive to. Okay, so but so far I'm glad that I have not. And it's been out for 3 months. I haven't heard of any adverse effects or anything like that. I've heard a lot of positive effects that can't even be explained by telomeres. Okay, so but when I say it's a fraction of a plant extract, it still has 10,000 plus molecules in there. So there's some other benefits in there that we don't know about.
I want to ask you, and if it's controversial. You don't have to go into it, but because there's so many schools of thought, right? There's the Horvath clock and Horvath's method of biological age, and they're looking at, because they're incorporating not just telomeres, they're looking at other things. Maybe the age of your liver is different than this. From what we do, my company, the only measure that we have is methylation. So, it's
you're measuring methylation. I did not know that, but I will tell you, that's probably the best way to measure your aging right now.
Thank you.
Okay? I mean, but even though but nobody knows how the methylation is controlled. Okay. Right. Nobody knows why do these methyl groups disappear or appear or whatever. Some cases it disappears, some cases it appears. What's causing that? Mm-hmm. Well, the explanation is telomeres. Okay. The telomeres are controlling the dimmer switches on the methylase genes.
Got it.
That are controlling that, and also the genes that control where the methylation goes. Okay, so we've actually done experiments here where we have in vitro, where we have allowed telomeres to get long, short, long, short, long, short, and we find the methylation patterns change just proportionally. So, I mean, we haven't published that or anything like that. I'm waiting for other. We don't have a publisher perish mentality. Yeah. And it's a lot of work to publish when we have so much science to do. Sure. But I tell everybody that, and I'm hoping that somebody else will go ahead and publish that, because I think that's really critical. But
it's critical because now you have a 1-to-1 ratio of telomeres to methylation, and it makes total sense. So now you can go in and measure telomere length and align that with epigenetic expression related to aging.
The problem is that measuring telomeres is not that easy.
Right.
I'm the inventor or co-inventor of every method. I can manipulate them with slight changes to make the telomere lengths whatever I want them to be.
Yeah.
And the only company that actually does measure telomere lengths right now that I think does a really good job is a company called LifeLength, LIFELENGTH. They're in Spain. And that's because they measure the percent of telomeres that are short, which I think is more important than measuring the average telomere length. It gives you more accuracy. But now, DNA methylation combined with other things, so my favorite company for measuring aging is actually, what do you call it? I'm going blank on their name, but I'll think of it in a second. But they do not just DNA methylation, they also do other things too. I don't think they do too much of telomere length because they know how difficult it is and how inaccurate. Yeah. But I do believe that telomeres are still the kingpin.
Yeah.
It's just
that's what we did methylation because it's easier to measure than
true diagnostic.
True diagnostic. Yeah. I've done their test as well.
Ryan Smith.
I know Ryan.
It gets better and better and better. But one of the key things is methylation. And so
So I want to ask you this question related to I already mentioned one person's name, Dr. David Sinclair. So I'll just continue this whole notion of, okay, he's in agreement with telomeres, and there's different schools of thought, but then he talks and triples down on senescence, on the zombie cells, and you're using all this energy in your mitochondria, and you deplete it to be able to. I'm not going to explain it as well as you can explain it. I'm just trying to paraphrase what Dr. Sinclair said. So I want to get your thoughts on that method, and there's other methods of people coming out and talking about aging. Is it all these things combined, or is it like we know it's telomeres and everything else is secondary?
Okay, so the problem with senolytics, the things that kill senescent cells, remove senescent cells.
Yeah.
I mean, that might be okay in mice, but in humans, because we have the telomere shortening, every time, so think of a 90-year-old person, probably 90% of their skin cells are senescent. If you kill all those senescent cells, you kill the person. Okay, that's just like catching on fire and burning 90% of your skin. Okay, you can't survive that. But senolytics is something that I don't do personally, because I'm worried that it's going to accelerate my aging. And because when I still have a few senescent cells, when you kill a senescent cell, another cell has to divide to replace that position.
Right.
And that causes telomere shortening. So I believe that Senolytics would be really, really great if it was combined with something like TeloVital. Okay. So now, because there you are increasing the chances of the cells being able to replace the killed senescent cells. Now, this is going to bring me into another tangent for a second here, but why this is really important is because, let's say you're that 90-year-old person and you have 90% of your cells are senescent. Well, all of those cells have critically short telomeres, okay? And which means they're almost at the point of dying and they can't get any shorter. Okay, well, we and others have shown, it's well published, that when telomeres get really short, they become a lot easier to lengthen. Okay. And so, and the analogy that I've come up with to explain this, because nobody has an explanation for this really, is that telomeres are not a linear piece of DNA. They're like a ball of yarn. And the tip of the telomere is inside that ball of yarn. So when telomerase comes along and wants to lengthen the telomere, it can't get to the tip of it because it's buried inside that ball of yarn. But when the telomere gets really short, that ball of yarn gets smaller and telomerase can get into it easier and lengthen. And so something like TeloVital is expected to be able to lengthen the shortest telomeres, the critically short telomeres, even though the longer ones will get shorter. So that's a key reason, because even the cell that's going to divide to replace that senescent cell, that also has short telomeres. But TeloVital might keep those cells from getting shorter telomeres. So anybody who's taken a senolytic should also be taking TeloVital. Right. And I'm going to say there are other companies that have products that say that they produce telomerase. Okay. But the problem is none of them actually demonstrate telomerase expression. What they do is they show that before the person takes the supplement or treatment or whatever, or diet or lifestyle, beforehand their telomeres are one length, and then afterwards they're longer. Okay, that's a common misconception that even Dr. Calvin Harley explained to me when I was still at Geron, is that you can lengthen telomeres in your blood. I don't want to use the word lengthen. You can make it so that after treatment, the telomeres are longer than before treatment without lengthening. Mm-hmm. Because you can, if the treatment is at all toxic, cells with short telomeres are more sensitive to toxicity and they'll be killed off. Therefore, the average length of your telomeres of the cells that remain will be longer. And that's what makes the measurement things wrong, because they think the telomeres are getting longer, but they're not. The analogy that I like here, I always filled with analogies, is if you have a company and you fire your dumbest employees, the average IQ of your company goes up without anybody getting any smarter. Okay, the same thing's happening in our blood. You kill off the cells that have short telomeres, the average length of your telomeres go up without any lengthening. Now, the other side of the coin is if your product is at all immunogenic, it will induce like naive immune cells or memory cells from young ages to divide and infiltrate the blood to fight whatever thinks is invading the blood. And these cells that come from your bone marrow and stuff are going to have longer telomeres because they have not gone through very many cell divisions. So they enter the blood and suddenly your average length of your telomeres are longer without any lengthening. And the same thing is, the analogy is, if that same company you fire the dumb employees, if you hired a bunch of geniuses, the average IQ of your company goes up without any lengthening.
Yeah, I'm gonna get a call from HR about my dumb employees.
Well,
I want to ask you one more question about this. So is it, if somebody's in their 70s and they realize, they live this fairly healthy life, they did some exercise, they ate fairly well. Now they want to go forward and work on extending their health span and their lifespan. Can they start now, or
Oh
is it
yeah,
already?
absolutely. That's a great age. In fact, all ages are going to be really good to start doing anything to lengthen your telomeres
Yeah.
to enough to decrease the rate of the shortening. And the shorter your telomere, the older you are. I don't like the word older, and I just said it. The longer lived you are,
Longer lived.
the shorter your average telomeres are, and so the greater chance that you are going to actually
Yeah.
get some
It's
age
the
reversal.
ball of yarn example that you just
Yeah, yeah.
mentioned.
You're
Yeah.
going to get some age reversal by lengthening those critically short telomeres. And that's the only way I've been able to explain why TeloVital has been getting a lot of people claiming that they're seeing age reversal. Okay.
Mhm.
More people than you would imagine from just placebo effects or anecdotal effects or something like that. And I've seen some of these things and they're real. Okay. So I can't, I'm a scientist. I'm not going to say, hey, here's a lot of anecdotal data that says you should take this product. But there is reason to suspect why this anecdotal data might be real.
Oh
Okay.
yeah,
And that's
for
by
sure.
lengthening the shortest telomeres. But I do believe, I was saying that no other company that I can think of at the moment.
Mm.
In fact, I don't want to blast any other companies. Okay. But I would say that if somebody wants to know if a other product really does lengthen telomeres, they should ask that company, do they have data showing that their product induces the production of telomerase? Because if it doesn't, it does not lengthen telomeres. The only way to lengthen telomeres is with the enzyme telomerase. And so they might be just seeing longer telomeres because of what I was talking about, toxic ingredients or immunogenic ingredients causing telomeres to appear longer. But they didn't lengthen the telomeres. And as a result of everything, when you add new cells to the blood that have longer telomeres or get rid of old cells with short telomeres, you have to have cell division to either replace those killed cells, or you have to have cell division to get those cells into the blood, the long telomere cells into the blood to begin with. And that accelerates aging because it required cell division. So even though they're claiming their products reverse aging, they're probably accelerating aging unless they can show you data that shows that their product produces telomerase.
How do you measure telomerase? Is that a blood test or is that
Easy.
a
Yeah, blood test is, I'm again the inventor or co-inventor of every method. Okay.
Great.
And it's easier than measuring telomere lengths. Okay.
Got it.
But yeah, you can measure telomerase. And so the question is, when somebody says, no, we didn't do that, we don't have data showing that it induces telomerase. The question is, why not? Since it's easy to do. I mean, it tells me that they tried it and it didn't work.
Sick.
Okay. And so they didn't understand why it didn't work, but they saw longer telomeres. So they're going to say that their product lengthens telomeres. But that's what I would say, that if you go to a company and their product it says that they lengthen telomeres and they can't show telomerase, walk away. That's why I did so much work trying to develop TeloVital, or discover the ingredients that are in TeloVital, was because I wanted to find something that really did lengthen telomeres and was a plant extract. So it didn't require 25 years of FDA approval like gene therapy is. That's what it's all about. But I recommend people take TeloVital. I think it's the absolute best thing anybody can do to slow down the rate of their aging.
Give me sort of your crystal ball thought in the next 20 years with the acceleration of the couple things, with the acceleration of gene therapy, the acceleration of the research that you're doing, and also the introduction of machine learning, because it allows you to exponentially, achieve some of these things quicker in terms of even analyzing the research that you're doing. What do you think the next 20 years holds for, longevity and health span?
Okay. Well, I believe gene therapy will have a major impact in clinical studies, and also the really wealthy that can afford it.
Yeah.
Because it's very expensive to do this gene therapy. And like in the clinical protocols I've written to follow exactly that clinical protocol, it cost me $1 million to treat one person once. Now, BioViva is doing some things that might make it a little less expensive and stuff like that, but it's something that if I was a multi-billionaire, I'd be banging down BioViva's door and saying, treat me. Okay. Because I'm very optimistic about what they're going to do, but it's not, the general public's not going to be able to take it. I'm of the mind that I want to, when I have something that lengthens telomeres, I want to drop it from airplanes.
Right.
Okay. And give it to everybody. But so I believe that in our continuing research, we will discover more potent telomerase inducers from plant extracts. I've said we've only tested 20,000. There's 3 million different plant extracts out there, and then we can make fractions of all those. So we got a long way to go. We can do this. The problem is it costs us $2 million a month to do this screening. Okay, but we do as much as our screening as our funding allows us to do. But I think that I would say that before gene therapy ever becomes a thing that every single person can use, I believe that we will have a pill that you can take that will do just as good a job as the gene therapy. But it's going to take a while. And I estimate that it would take 3 years if we could get the funding that we needed to continue I'm not trying to make a pitch right now, but if we could get $2 million a month to spend on the research, we would have a pill that would actually cure the aging process in 3 years from now. And I'm hoping that we get that money from Touchstone Essentials, because we get a royalty that goes straight into our research from TeloVital sales. Again, I'm not making a pitch that people go buy TeloVital to get us research funding, but there is a icing on the cake. By taking TeloVital, you decrease your rate of aging, but you also increase our research to allow us to produce more potent things. And believe me, after working with Touchstone as long as I have now, there's no other company that's going to be marketing these products when we develop them. Okay, so Touchstone is a company that I think people should really hook up with. And let me just say, I've done critical meta-analysis, due diligence, everything on their other products too. They have products that nobody else has, products that turn sugar into fiber. Sugar is bad for you, fiber is good for you.
Yeah.
Glucose control, I take that. I highly recommend. I don't know anybody else who sells something like that. There's things that get rid of toxins that they have, and that are kind of going blank on the name of the chemical. It's not a chemical, but it's a mineral kind of thing. But there's chemicals in it. It's called oh my God, I'm embarrassed that I can't remember the name. But maybe you can put something in the thing that nobody else has. These things really get rid of.
I'm just surprised that people like Elon Musk and et cetera aren't investing like hundreds of millions of dollars into this technology and into these companies to be able to.
They are, but what they are also doing is they're taking control of the science. And I always say this is why cancer is not cured yet. Okay, is because investors come and put a whole lot of money into the company and then take over the company. I had that experience. I raised $60 million when I first started this company from 50 plus investors. And the first thing, and so I thought, okay, we're going to have aging cured in just a year, 1 to 3 years. And then investors just came into our strategy meetings, told us to stop everything we're doing and work on other things to make them quick return on investments. And so we did that, and we were successful at doing that. And when we were successful at doing that, they said, okay, now let's sell the company and all of Bill's IP, and we'll make a lot of money. And so I was lucky. I had 2 of the investors that just stood up for me and bought out all the other investors that just were in it for the profits. And they turned 100% of the company over to me, and all they want now is cure for their aging. And believe me, the second I have it, they're getting it.
Yeah.
Okay. And because there's a few people out there that are more interested in the humanitarian return on investment than the financial return on investment.
Love that.
And that's the problem with all these known billionaires.
Yeah.
Well-known billionaires. They all seem to be driven by profits, but that's how they got where they are. Okay. And so I'm looking for unknown, ultra high net worth individuals that want a cure for their aging, that would just come in, provide the funding, take care of me and my scientists, allow us to do our science without any distractions or anything like that. And we could have their aging cured within 3 years. And when I say 3 years, when we extrapolate from the results we've already had, it's actually 1 to 3 years.
Right.
We could get the $2 million a month funding that we need. We could have a pill that cures aging in 1 to 3 years. And if it's a plant extract, we won't have to do clinical studies. If it's a pharmaceutical, we'll have to probably do some clinical studies. But I have a feeling, I just have this feeling that if we have a drug, even this pharmaceutical drug, if we have a drug that turns somebody, like I would have loved it if Betty White was still alive, turned her into a 25-year-old. And she walked out on stage and everybody knew it was her. And she looked 25, felt 25, and behaved 25. What's the FDA going to do? I mean, we could put that on.
They're going to make you still go through the 3 phases.
We just have to build an airport on a small island in the middle of the Pacific.
Exactly.
Everybody's going to go there to get treated with that pill. That's what but the pharmaceutical is a long ways away, and it's going to be battled with kind of all kinds of obstructions.
It's just the way it is in the US. I mean, even stem cell therapy, like we're so behind. You have to go offshore and all these other things to do, instead of saying, all right, let's just make everything available and put a black box warning on it that there will be, like you said, everything has an adverse effect, but give it to the person to make a decision instead of Big Brother making a decision for you.
Let me make another plug. Elizabeth Parrish, the president, CEO of BioViva.
Yeah.
She did her MBA on something that she called best choice medicine, which is doing exactly what you're talking about. And it'd be a division of the FDA where people who, especially suffering from life-threatening diseases or diseases that make life not worth living, they would get a pass.
Yeah, it's a waiver. Exactly.
A waiver to do whatever they want. Okay. And it's called Best Choice Medicine. She and I co-authored a paper on it, and people can find it on the web. And we also, there's YouTube videos of me talking about this. And it's not telomeres, but it is definitely, I'm only interested in people being happier and healthier, period.
Yeah.
And I think that what you just mentioned is exactly what we need to be doing. And there's other things, but there's actually, I'm a member of the Coalition for Radical Life Extension. I'm also a member of the Transhumanist Party. And all these things are towards trying to make people happier and healthier. But the Coalition of Radical Life Extension is organizing a march in Washington, D.C. in September, I think, of 2026 to make a pitch that the FDA starts doing exactly what you just said.
Yeah. I mean, it makes total sense because, and I'll tell you, and I'm going to shift focus to a couple of questions I ask all my guests, but, and this isn't a plug for me, but we had, I worked with a group of doctors where we treated cancer patients with phytocannabinoids and end of life cancer patients. They were given 6 months, less than a year to live, and not FDA approved. Schedule 1, I can tell you, we extended people's lives from 6 months to 6 years, gave them quality of life. It's their choice. They're adults. They're able to do what they want to do. We didn't say we were going to make any claims, we're curing anything. We didn't do it in the scientific, but anecdotal evidence displayed right there with plant medicine and being able to provide a lifestyle that's beyond what, chemo and radiation, all the other stuff that they would be going through. So, I mean, it makes total sense. And they're adults enough. You're an adult. You make a decision what to do with your own body. So I completely agree. And I'm on board with this movement as well.
I've seen some of those studies and they look good. I got to tell you that I've invented many cancer drugs, okay, and taken them through clinical studies. And they work perfectly in mice, but they failed in humans. And some of them are like Rytelo, which is one that I invented. It's actually been approved by the FDA. But I can tell you a lot of people in the placebo control group would have their cancers cured just because they believed they were getting the real drug. And so it's hard to really show that something is actually curing cancer or not just providing some psychological benefit to you.
You know what's the difference? Because at the end of the day, if you're
right.
Right. If you're depressed, and I've had this, like, once again, I'm not a doctor, I'm not a scientist. I'm just a sciencey person who's interested in this. And I know a little bit about genetics, very little, but I have a company. But if I've had this happen so many times where somebody was diagnosed with cancer and a week, 2 weeks later, they get shingles. And if you start looking at this whole expression of your body wants to cure, and if you are stressed, you talked about stress as number one. Like, if you're able to alleviate that and you're able to do it with phytocannabinoids while, maybe reducing some inflammation and making them feel better, now your mindset is, I want to heal. And you're, doing everything. Your body's actually trying to heal itself. So does it matter?
That's why I said the studies were good.
Yeah.
Before I started talking about people getting, because you're right. It is better. It is working somehow.
Yeah.
We don't know the mechanism of action, and whatever, but it's making it easier for our immune system, let's say, to kill the cancer cells.
Right.
Who knows? But I can tell you, there's like 70 different cannabinoids that we tested here at Sierra Sciences, because we sure wanted to find out that one of those would induce telomerase.
Yeah.
And none of them did. Okay, which was really disappointing. Okay.
Well, can I say, have you tried using the combination of the cannabinoids and the terpenes with it instead of the isolating of the individual cannabinoids? Because I believe it's an effect that has multiple, because you have different receptor affinity, and you have the agonists that come with it from the terpenes as well.
Okay, so we have done a lot of synergy studies, but it's because it's so expensive and stuff like that. We only do the synergy on the hits that we have to see if we get more, but we haven't tried really synergy on two things that were negative. Okay, but send me a list of the things that you want us to test.
Okay,
and we'll work out some kind of deal with you. Okay, and we'll test them.
All
okay,
right. Let's do it together. I'm in. I'd love to participate.
You have any of your members of your audience that would be willing to put up the $2 million a month that it takes to do the screening?
Let's go.
That's about a month's worth of screening.
That's what we're talking about. Everybody, whoever's got money, send it to Dr. Bill.
Yeah, but put a clause in there that says that I won't use it to buy myself a big yacht.
Exactly.
Because that's not me. But feel safe when you do it, because I'm every penny I make goes right into the research here.
Yeah, for sure. Bill, I wanted to ask you a few questions that I ask all my guests. They're more music related questions, so nothing to do with science. We can shift our brain a little bit. If you recall, what was the very first concert that you attended?
The Doors in high school in the late 1960s.
Oh.
And it just blew me away. My very first song, I know you didn't ask this, but when you started asking that, I remember the very first song that I ever liked was Downtown by Petula Clark. I heard that song, I became somebody that liked music. But before then, I hated music. I was maybe 11 years old at the time or something like that. But I just, people would turn on music. I say, turn it off, turn it off. Then when I heard Downtown by Petula Clark, I just became a fan. When I went to the Doors concert, I did enjoy it a lot, but I had a problem and a lot of people have this problem. Like the bass, the drummers, or whatever, something causes a vibration.
Yeah.
That makes a lot of people actually get nauseous, sick to their stomach, and you can feel your heart moving and stuff like that, and it becomes extremely uncomfortable. And I have had to walk out of concerts just because of that kind of thing. And I've then Googled.
Maybe it's too loud. Maybe it's too loud and it's affecting your ears.
It's actually it's only during certain parts when all of a sudden some boom, 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 boom causes a vibration. It's suddenly I guess the cells in my heart and my stomach are at the same vibration, and it causes them to vibrate just like a tuning fork does.
Interesting.
So I don't go to concerts anymore.
All right. Well, the next question was, what was the last concert you attended? So was that the Doors?
No.
First and last?
I did go to a few more. God, what was it? It wasn't Beatles. I never went to a Beatles concert, but Beach Boys. And they did not have that kind of music that caused.
I was going to say it wouldn't be a lot of bass in the Beach Boys. All right, one more music question. Let's see how we do with this one. For the next year, you could only listen to 5 albums. Now, you don't have to remember the name of the album. It could be a Beach Boys or Beatles album, whatever, Doors. What would be those 5?
Rebecca McEntire. I don't know the name of the albums, but Taylor Swift. I'm a really big Taylor Swift fan. And I don't remember the name of her albums. Who's the Korean guys, the 5 guys that oh God, I can't remember their names. I'm terrible at remembering names. This is not an age-related thing. I've always been terrible, but there's the I can almost hum the tunes, but you don't want to hear me hum. But there's, let's see, I see, who else? God, how many did you say I have to come up with?
5.
5. Okay, I'm going blank. I know the people, but I can't remember the names.
Like you're talking about BTS, like that.
Yeah, BTS.
Oh, okay, got it.
BTS,
Okay.
yes. Okay, were you figuring that one out with a Google search or something?
Well, I was just trying to remember. I'm
They
like,
are
oh
fantastic.
shit. Yeah.
I just love watching them.
Got it.
But it's not just listening to music, also watching them perform. But there are some really good singers that I just absolutely like, and I can't remember their names. But if I thought about it, can I send you a text and tell you what they are?
Sure, no problem.
Yeah.
Whoever comes to mind. Okay, so final question for you. Please describe what your room looked like when you were growing up.
Oh, it was always a mess. I was always messy, and it still is. I mean, it's like, thank God for being able to hire other people to clean up after you. But I just remembered another song. Who's another person that I really like, and I can't going blank on her name. But she was dating the guy that played Thor, or the brother of the guy who played Thor. It's weird ways I have of recalling things, but that's the way my mind works. And she was a teen singer, and she's now an adult. But God, what's her name? And she sang, "I can buy myself roses. I can buy my own roses. I can sing my own song." Who's that? Do you know who I'm talking about?
You mean Miley Cyrus?
Yes, I love her. She's just fantastic. Everything she ever sings, I just love. Okay, I don't know why I can't remember their names, but I never can. And it's like in my whole life, I've never been able to remember it. But I do remember the songs, and I love listening to music.
That's good enough. Where can people engage with you? Where can they get more of your content? I mean, you gave out so much information. Is there anywhere that people can go and
this is going to sound odd. Since I'm just a scientific company, I'm not a marketing company. I don't even have a business part of the company. Everything's science. We don't really do a very good job of keeping up our website. I've just asked some people just recently to set it up, and it's completely down right now. But I think the best place to go is actually Touchstone Essentials website. Okay, they've put a lot of stuff about me because they're kind of endorsing the TeloVital, it's pronounced, more on who invented it than what it actually is and stuff like that. And so they've got a lot on me, but their website is www.thegoodinside.com. And you go there and you're gonna find a lot of products. I'm still angry at myself because I can't remember the name of the thing that gets rid of lead and other things. I wish somebody would text you or something like that and tell you, and so you can tell me what it is. But they also have like fulvic acid, which is the active ingredient in Shilajit. Shilajit is great. Everybody knows Shilajit is great, but Shilajit has side effects. Fulvic acid is the active ingredient and doesn't have any side effects. Boy, I got a few things to text you. All right. But people can go to their site and they can see exactly what I'm talking about, and then they can call me up or email me or something like that.
No, we'll do a part 2 at some point. You'll get all this stuff, and then when you have your invention, you have your $200 billion grant, then we will be there to get you back on the show.
Please give out my email address. I'm a person that loves to get email. I mean, when you're working in the lab all day long and you need a break, nothing better than reading emails and answering them. Okay, I love doing that. So my email address is B as in boy, Andrews at Sierra Sci, and that's spelled SIERRASCI.com. Make that available. I'd love to hear from people.
Great.
Because I'm all about making people happier and healthier, as I said. I love living, and I hate thinking living is going to be any less lovable for anybody because of this dumb thing called aging.
I love it. I love what you're doing. And for my audience, thank you so much for listening. Thank you so much sticking with it. If you're here, then I guess you like what you heard. So don't forget to like, subscribe, share. Thank you for the wonderful reviews. They've been fantastic, and I really enjoy reading those as well. Thank you for buying my book, Making Cannabis Personal, and my new book is coming out shortly. It will be about music, so nothing to do with cannabis or genetics. And I just want to say thank you again so much for being on. Really, really appreciate it. And the work that you're doing is fantastic.
Right. Thank you. Thank you for letting me talk. I love talking about this stuff. All right.
You can tell. Hey, everyone. So does anybody know what greening out means? I've been the recipient of so many calls over the years of people greening out. That means they took too much THC and had an adverse reaction. So I always recommend other things, but nothing really concrete. Now I can. There's a solution. It is called Antidose. Antidose.com is quick. It's rapid. It works immediately, safe, and effective. So I love my props. If you are toasty or you are burnt, that's the difference. Now, I also love the 1 gummy, 2 gummy, 3 gummy floor. So just be aware, very narrow therapeutic window. You know exactly what you can take now if you've taken too much. Once again, it's Antidose at antidose.com. Get yours today. Hey everyone, thank you for watching and listening. And don't forget to hit the subscribe button, share this episode, and hit that bell. Like, comment. I so appreciate that. And check me out on social under Len May or Len May DNA. Find out more on LinkedIn or lenmaycoaching.com. And remember, everything is personal.